Hello and welcome to Nikolai's genetics lessons, and the first question of the day. The genetic code is essentially the same for all organisms. From this, one can logically assume which of the following statements to be true. Before we will choose the correct answer, I want you to take a look at this table, and this table specify codons which specify amino acids, and these codons we can find in the messenger RNA. The same amino acid, for example phenylalanine, can be specified by different trinucleotides or codons, and leucine, as you see, specified by 6 different variants. Many of you probably from your textbooks and from the literature have impression that this codon table is universal, but this is not quite so. I want to show you some exclusion from this rule. Here is example number 1. As you know, AUG specifies start codon, as I just told you earlier, but there are also alternative initiation codons. In rare cases, translation in eukaryotes can be initiated from the codons other than AUG. A well-documented case, including direct protein sequencing, is the GUG, start of the ribosomal P protein of the fungus. So this is going to be the first example, and here's the scientific name of this fungus. Second example would be vertebrate mitochondrial code, and in mitochondria a number of codons specify different amino acid than in standard trinucleotide sequence or standard sequence table. For example, AGA in standard specify arginine, but in mitochondria specify tyrosine. And for example, AUA specify methionine, and in standard isoleucine. So as you see, our mitochondria has slightly different codon sequence, or the same codon sequence specify different amino acids in mitochondria. Here's yet another example of the yeast mitochondrial code, where we can find that 6 trinucleotides or codons specify different amino acids from the standard table. And here's the scientific names of the yeast in which mitochondria has non-standard codons, which specify different amino acids if we compare with the standard trinucleotide sequence or codon sequence. Here is another example: mold, protozoan, and mitochondrial code of the mycoplasma and spiroplasma code. And you see that, for example, tryptophan would be specified by codon UGA, and in standard codon table sequence UGA specify stop codon. Now let's return to our first slide, and now you see that this codon table is not universal but nearly universal, because there are actually many more examples of exclusion from this rule. There are about 42 more examples of different clades and families where we can find such small differences, but we can say that codon table, this sequence of the trinucleotides, not universal but nearly universal. So true in 99.99%, but still there are some exclusions exist. So now let's check variants of the answers. Variant A: the same codons in different organisms translate into different amino acids. This is not true. Those, as you know, small exclusions exist, but generally would translate into the same amino acids. So different organisms have different types of amino acids. This is also not true. And DNA was the first micromolecule. This is also not true. But the last variant, a gene from an organism can theoretically be expressed by any other organism. This is true, but also you have to pay attention to the word theoretically. Because, for example, if we'll take a gene from the human and would insert it into the bacteria, bacteria wouldn't be able to express it. Why? Because our genes have introns, and bacteria genome doesn't have introns, or nearly doesn't have introns. So in order for the bacteria to express human gene, such gene have to be modified and introns have to be removed. And it is better to express human gene, for example, not in bacteria, but in some eukaryotic organism, for example, in yeast. And next question, which of the following processes occurs during transcription? Here is double-stranded DNA, and here is a messenger RNA. When messenger RNA produced, we call this process transcription. And when this messenger RNA is produced, large and small ribosomal subunits would attach during the process, which we call translation. This messenger RNA would be translated into the sequence of the amino acids, into the polypeptide, which would then later would detach from the ribosome and would make three-dimensional form, which we call protein. So this process we call transcription, and this process we call translation. Now let's check variants. Variant A, messenger RNA attached to the ribosome. This is not transcription, this is translation. Proteins are synthesized. Again, this is translation and not transcription. RNA is synthesized, and this time this is correct statement. This is example of transcription. And the last variant, DNA is replicated, is completely out of this picture. So the correct answer, C. And this is all for today. Subscribe and see you in the next video. Goodbye.